Okay, the recording has started. So now let's resolve this exercise. So as I mentioned before, we have two exercises. One of them will be to calculate the differential expression with CAM-R-band, and the second one will be with DSEC. So we were using this data. I already presented that in the first part, so I'm not going to go there. So the thing that I'm going to start to do it is just going here. So first you need to transfer with the string type the output of the server of the files that you want to analyze. So in my case, I have been doing that, and I'm here, no? So in my case, I create this directory. Well, I create the directory differential expression, and then here I have two, string type CAM-R-band, and then I2TA2020 count. So here in Valgo, I have all the different files. So you can see that all the files has the sample names, no? I2TA2020 rep1, rep2, rep3, and so on. So this is my directory. Okay, the directory where I copy everything. I also have here this pheno data.txt. One thing that I mentioned is, please keep in mind that because I made a mistake, okay, in the file names. For C24 per Columbia, this X is lowercase, and for the other one, the X is uppercase. And I know that already that gives you some problems, so I would like to apologize for that. Okay, so let's go back to this directory. So as I have here the Valgo. So that will be the first part, just preparing the files. The second, open RStudio, okay, and then we want to upload the libraries Valgo, every color viewer, gene filter, and Tabular. So let me copy this. And as I said, I want to go to library. Well, I have two options. One of them is going to library, I copy that. The other is you can go here to packages, and here click Valgo. It's already there. The other is Tabular. That is already here. Yeah, sorry, I don't have R-color viewer and gene filter. So gene filter, DEFG, gene filter is uploaded, and R, last one is R-color viewer. So that would be another option. You can just click in this and that will give you the files, okay? We'll upload the libraries. As I said, you can say library, and will be, for example, we want to use CAM-R-band. CAM-R-band. One moment. No, sorry, we are working with Valgo. Apologies. Yeah. Okay, so that's an example. Also, one thing that is important if you are not working with RStudio is that when you are writing things in RStudio, you have the option to, once you are writing things, you have here different directories, environment, history, connections. So in history, it's going to appear the commands that you are writing. If for some reason you want to keep these commands, my recommendation is you open here a new R script, okay, and then you click here and you move to source, okay, and then you will have in a file this. So here, for example, you can store this. Say that you want to store this here in a string type, like R commands for I2TA2020. Okay. And as I said, you can do this with everything. So with the library DeepClear. Is DeepClear or DeepClear? I think that is DeepClear. DeepClear and Color Brewer and DeepClear are Color Brewer and Gene filter. Okay, as you can see, I have them here. I want to pass to my command. Okay, perfect. Okay, so that is the previous step. And then the last one is to prepare a tabular file, data, with experimental design. Okay, so potentially this is the files, and then you want to put all these different things. So you want to have a sample ID in IDs, accession name, type, and replicate. So you have two options. You always can open a text file in text edit and write it everything. But my recommendation also with the file is because you may have the problem that the file names may not be the same. Like in my case, you don't class yourself, like I don't class in my typing. You always can use the function file dir file. Dir file. Sorry. Again, is something that I have here. List files. Okay. So it will be list files. And then it will list the files that you want. Of course, I'm not in the right directory. So if you want to move to the right directory, now when you are in the right directory, you always can go here and click set as a working directory. Okay, I want to keep this here. This is important. And then again, if you type list files, in this case, I want to list the files that are inside Valgo. That will give me the list of files. And of course, sometimes it's just more easy to use those. Of course, also you can create as you did with the dissect part, you can create this data table manually if you want here. Okay, that would be a possibility too. Okay, so that is thinking in setting up the system. So let's start with the data upload. For data upload, first we need to upload the data. So in that case, we need to just use read the limb to do that. So if I do this, keep in mind always that copy and paste mess up with the quotes. So I do this and now I run it. So I have data here. You will can test how they look like. So this is my data. Then next will be to sort, because sometimes that gives you problems. So I need to sort. I need to sort them. Okay, so I'm ordering by IDs. So if I run this, okay, it will not change how they look like here. But when I was preparing the exercises, this gives me problems. Okay, so that is the second part, and then we go to the most important part, that is to upload the directory with Valgo. Okay, so copy and paste. So several things about this. So how works this? So the first is I'm telling this data there that it need to look inside the directory Valgo. Remember that I have all these directories, and inside Valgo is when I have all the samples. So I'm telling Valgo function to go and look into this directory to retrieve the samples. Then the thing that I need to look is for sample part and I2TA. So if for some kind of reason I have seen here that doesn't start with I2TA, this will ignore those. That can be convenient. And then for the data, I will upload phenodata. That is the one that I'm here. So if I run this, it will start to upload things. Let's suppose that you have problems. Let's suppose that in some ways you are not uploading the right phenodata. So one option that you always have is to upload only the first two, data there and sample pattern. See if that works. If that works, that you know that the problem is in phenodata. Okay. If that doesn't work, you know that the problem is in these directories already. So that would be a good way also to just trying to discover or check what could be the problem. Okay. So it's in case of troubleshooting, because I saw that some of you have that problem in the chat. Okay, so we have already the data here, almost. So once we get uploaded data, we can take a quick look of what is there. It's using just the name of the object or just summary. So if I type the name of the object, it's telling me that I have 74,240 transcripts and 12 samples. And then, of course, I can use, I'm going to use head, okay? But you also have the gene expression function that is going to retrieve the expression of the genes. So for example, if I do this, it's going to apply gene expression on the Valgo object. I'm not sure what is taking so long. Okay, so this is how looks the first 6 lines. Remember the head with prints only the first 6 lines. So most of them looks like an empty expression. Of course, we are not interested to know just the first lines, so we are more interested to know what is going on with everything. So you always can type summary, and that will give you a summary of everything. So this go and then summary expression. Oh, sorry. Well, indeed, I don't need to do this. I can always go here and move. Okay. So with summary, it's going to give you some stats. So for example, the stats that you can see here are things like mean, maximum, minimum, and so on. So minimum, you can see a lot of 0 values that we will need to filter out. In the mean, you see that the mean is more or less the same, 52, 42, 50, 53, 44, 55. So at this point, looks like everything is fine in terms of the genes. And then you have these maximums that looks more or less homogeneous too. We can look for the summary, or also we can look, that is summary of the genes, but also we can test summary of the transcripts. That will be this function that you can see here. And again, it's more or less the same. Because we are looking for transcripts, not for genes, and the expression in this case, the mean is going to be lower, okay? Because it's not the sum all of them. Okay, so that is in terms of, so I can put here, of course, if you want, I can share with you exercise 1.1. Yes. I think so. Yeah. Okay. So that is getting some stats. The next is the data filter. As I said, you don't want to have 0 values for everything. So one possibility is to just filter those out. So you can filter those out just with this command. Let's go here. Let's call this exercise 1, 2. And the thing that we want to run is this. Let's check that we have the right quotes. So it's a subset using this expression is why you have the expression between quotes. And we want to say that the genome subset is true. Okay. So we do this. We will be selecting. So again, we can run a BG to check. And it's now telling us that we have still 74,240 transcripts. And then if we want to type, for example, these commands again, it may give us slightly different. It doesn't mean that you still don't have minimums, because maybe you will have some minimum of 0 for some of the files, but not for the other ones, okay? So, oh yes, sorry. Apologies. I was saying, oh, this is strange. Okay, no. Yeah. Is that. You're right. Okay, so you see that we reduced the number of transcripts. And now that we have reduced the number of transcripts, if we look for summary, yeah. You will see that now some of these values of the mean have changed, okay? So here, for example, the mean is 52. Now still, if we have 0 values, you see that the mean have increased a little. Okay, so that is with gene expression, and again we can do with T expression too. Okay. So, as an example, I will run one of those. What happens when you want to have just a subset of some samples, no? We saw that we may want to compare only some things, and we don't want to compare everything. So one option is just to filter by sample names, as you can see here. No, so in this case, we don't want to have raw values text more than one. We, for example, want to have only the one that take pure line for type. So if we run this, okay, now you see VG subset PLN. You see that we don't have 12 samples, but 6. Okay, so that will be a good way also. So now, for example, if we want to get, so which one do we have here? So if we want to know which one we do have here, with add, you can see directories, I think. So we see that we only have C24 of Columbia. So that means that if you want to check the differential expression between only these two conditions, that will be a good way to do it without count in the variance of other samples, okay? So that would be a good way to do it. Okay, so that is for the exercise 1.2. Let's go to the exercise 1.3.
Actually, can I ask you a question?
Yeah.
How do you decide what's the threshold for the filtering? In this case was 1 FPKM, but is it a general rule, or is this something that you decide from case to case?
Well, indeed, it changes depending on the person. I mean, depending on the person that is running the analysis. I use 1 FPKM because it's the thing that appears in one of the tutorials. I think that makes sense. But of course, to be completely sincere, you will need to go and check what is your distribution. Also because maybe your experimental design is done in a way that you want to analyze also low expressed transcripts. So in this case, maybe if 1 will not be a good way to do it, maybe it should be 0.1. I don't know. Or if you have too much noise, you want to increase this to 10. It depends. So yeah, in this case, I'm using 1 because I think that by default looks like a good value.
Okay, thanks.
Yeah. The other question that I can see here is the option genome subset equal false. So genome subset is when you are having activated genome subset as true, you are selecting rows, okay? Meanwhile, when you say genome subset equals false, you are selecting columns. So you need to visualize how we have the data here. You have the genes in rows or the conditions in columns. So you need to say genome subset false when you are selecting columns. Okay. Yes. Okay, so that is 1, 2. Quality control, and now is when we're starting to do more interesting stuff. So first, we want to retrieve this gene expression data that we have as a data frame, because we want to play with the data frame. So we copy, paste the command, and as I said, now we have we want to select everything as a data frame. So if I run this, now we type here gene expression, and you see that, yeah, this looks like a data frame. Yeah, remember that we are working with the filtered data. And as you can see here, we have ATG or some microRNAs already showing some expression. Okay, so that is for selecting, selecting that. Then we want to change the name of the columns, because maybe use these FPKMs makes the name much longer, and we want to simplify. We know that everything is at FPKMs, so we want to get rid of these FPKMs. So if we run this, well, let me run this first. So if we run this first, so we can use call names to retrieve the names of the columns, and then we can use GSAT to replace things. So we are replacing FPKMs dot by nothing. So if we do this, you can see that we are removing FPKMs from the names. We could remove, if you want, also ARTHA, because it's not giving any additional value either. But well, in this case, we just remove. Let's remove just that. Then we want to have colors. So I define this set of colors, more or less looking into the replicates will have a similar color, and I choose 4 series that are red, orange, salmon, and green. So let's go here. Let's remove this space. Okay. And then let's run this command. Okay, so at this point, we have colors 2, and we are ready to start to plot. No, sorry, we are not ready to start to plot. We also want to have some short names, because still maybe these names are very long for a plot. So we want to run this. And then now, yes, now is the moment that we finally plot everything with this box plot. For all of you that you didn't use box plots before, I really like box plot because it can show you a distribution, okay? So each of the items that you see in the box plot means a percentage of the samples. So, okay, I think that I did a mistake. Let me see. Again, yeah. Again, be careful with, well, you already know that, okay? Be careful with the quotes thing. Here. Okay. So now let me run this. Okay, so we will have here our box plot. You see, no? Selecting all these colors, the names are smaller, so everything fits. The important thing here is that we can see, as I mentioned before when I was presenting the exercise, all the distribution looks the same. So keep in mind that you have something like a normal distribution that more or less this box plots represent that. The white line in the middle represents the median, the mean, and then the box represents 75% of the data. And then you have O groups, okay? So we can see that more or less all the data has kind of the same similar distribution. We may have some slight changes in the average, but more or less, so far so good, everything looks fine. For the O groups, we can think that the O groups, again, looks more or less the same for all the samples here. So that would be a good way to say, okay, so we are in the right way, as we mentioned before. Okay, so that is for box plots. Then we want to compare the expression between replicates. So we are going to select, for example, the first and the second replicate of the first sample, and we want to compare that to see what happened. When you are comparing replicates, they should have, as you know, they should have a correlation value quite high, because otherwise means that the replicates are not replicates at all. Again, keep in mind that this is telling me that, man, you are not using the right quotes. Okay. I have this here, sorry. And then plot. This is the plot. I think that the quotes looks fine now. Let's go to the final. This will be outline, the correlation value, and the expression. Outline will add a line in the middle. Then we want to have estimate the correlation between X and Y, and the square, and then the legend that we want to put in the middle, in one corner. So run this. Okay, so this is representing the replicates. In theory, it looks like they are not so bad, but keep in mind that the axes here are different, okay? So even if all the samples look like they don't have changes, big changes, keep in mind that that is still normal because we are comparing different samples, okay? But of the same thing, it still should be fine, but this R square is really, really bad, okay? It's 0.188 is really bad. Okay, so potentially we could do this by pairs one by one, but we don't want to do it with everything. So we are going to do it just, we are going to create a matrix, a correlation matrix. And the correlation matrix is basically, we are creating an empty data frame here. As I mentioned before, we are putting some names, columns and rows, and then we are running this loop in which we are just going to fill this with correlation values. No? So we're increasing one here, we're increasing one here, and then we are getting the gene expression for each of these indexes and calculating the RS, and then we are filling this matrix that we created here with all these values in a loop way. And at some point, we also are creating a heap one. So we do this. This is how it looks, at least my heap map. If you have something slightly different, still is fine, don't worry. As I mentioned before, here is when we are starting to think that there is something wrong. Why? Because replicates go together, okay? We have one replicates and then we have replicates 2 and replicates 3, but replicates 1 goes together. That is bad. Okay, a replicate should be with their own sample or something similar to their own sample. But if they cluster together, it's because the technical effect is higher than the biological effect. So that is a problem. Why? Well, maybe is, as I mentioned before, we have several reasons why this is happening. No? So as I mentioned here in the presentation, maybe we have a wrong sample name. One suggestion, every time that you get something wrong, okay, at least it's the thing that I do with myself, it could be a batch effect. No, because this should be different enough to the biological samples should be on top of, the biological variation should be on top of the technical variation. So batch effects is when you have, batch effects can be a problem when there are not much differences between the biological samples, then could be a problem. But in this case, we are talking about very different accessions. So I would not think that batch effects will be the cause. It could be, it still could be. Indeed, well, we don't know if replicates 1 were made together. We know that they were weird in some ways because the samples was already processed, so that could be. But yeah, I would not think that it's a batch effect. The reason also that why it's not a batch effect is because, as I said, then you find some of the samples that looks the same. No, Colombian SIM24 looks the same for the replicate 3, so that is very weird. So yeah, the thing I wanted to say is when you find something that doesn't fit your expectations, at least the thing that I always do is, the first thing that I always think is I make a mistake. Okay, so it's the first thing that I do. Think I make a mistake. Okay, so because of that, the thing that I did was, okay, so maybe when I was downloading the data, I make a mistake and I didn't tag the labels okay. So the thing that I did was I went again to the NCBI, and then I went sample by sample. I took this run name. I'm looking to the equivalence that I have for the names that I changed later. And also, if you look into your sample, so you open your FASTQ files, you will see a sequence ID. So the sequence ID that you see is this one, the name of the run. So I was checking everything, and everything looks the same. The thing that was interesting is that the samples with 90 base pairs clustered together. So what Andrea is proposing, it could be a batch effect. Okay, it could be a batch effect. But also in this case, it looks like the batch effects is that they mix also the samples together. As I said, I don't know at this point what was happening here. I was tempting to write an email to the authors saying we are using your data for an exercise because I wanted to compare hybrids with pure lines, and we are finding these problems. Well, I may do it anyway. Okay, so that is in terms of this correlation matrix. One way also to look is not to look for the correlation matrix, but the distances between the samples. So the distances between the samples can be a function related with the correlation matrix. So we can just subtract 1, and then we need to scale the distances, and then we can plot them. Okay, so let's do this. We are going to plot this. So as I said, we scale, and then here we plot. As I mentioned before, this is not surprising. This is another way to see the same thing. Remember that the correlation matrix at the end is a distance matrix too. So we are putting this in a 2-dimensional space too, and we can see that R1s clustered together, R3 clustered together here. Then we have R2s that clustered together, so this one, and then this R3 clustered together with this one. So at the end, we have a really weird situation here that we still don't know what's happening. So at this point, okay. So at this point, if the last thing that remains was going to the paper, as I mentioned before, and look into the table that they supply in the supplementary data and check if we have the same number of reads, and we did. So for sure, we are sure at this point that was their mistake. Okay. If that things happens to you with your analysis, at this point is the point where you really should be worried, because that means that your RNA-seq analysis is not going to be good. Okay. Why? So remember that during the experimental design, I mentioned that things that you can do to avoid these kind of things is always, when you are extracting your RNA, separate your RNA into aliquots and use one of the aliquots to perform QRT-PCR of genes that you should know, what is the thing that you should get. And that will be a good way also to verify the identity of the sample before you send the sample for sequencing, okay? Because errors happens. So that will be for the differential expression. So, okay, so that is the quality control. At this point, still we need to continue the analysis. So the last thing that we did was we estimate the results. Okay, so we run this statistical test on genes with the covariate type. We want to have the fold change and FP-types. So exercise 1.4. Okay, so if we run this is when we are running the analysis. So now is the statistical test. And now we can type head and check what is in results genes. You can see we don't have expression. We only have the gene, the fold change, and then a p-value that tells us if it's significant or not. As you can see, the corrected p-value is bad like hell. Okay, so that is that is how we estimate this. In the exercise after this, I mentioned that we want to add the gene names to the output table. So I run these different commands because we want to have we want to create a table with the expression and then check the expression with that. Because I said you can see here that we don't have expression, so we can merge the data that we have. So to do that, several steps. Remember, okay. So several steps. First, we get a table with expression. Okay, we are looking for transcripts in this case. So yeah, I guess at this point it will be better not to have transcript, but sorry, genes. Probably, yeah. Okay. So we have a table with the genes and the expression of the genes. Then we want to have gene names, and then we want to merge gene names with the results of the differential expression. And yeah, for gene expression, we don't need to use all. So again, apologies. Okay. By must specify unique valid cause. Yeah, I guess that the problem is when I have changed just this quickly, I didn't realize that the columns from gene expression may not be the same. So So this is my table. Yeah, so the problem with my table is that I don't have IDs, so I will need to add an ID here. So to add an ID could be something like, call this BG table. I want to add an ID column. Indeed, I want to use as a join gene ID. Okay, so for gene names, so we have gene names. Okay. And we want to have result genes. Okay, so in this case, yeah, we can put here whatever name we want. So we can put here gene ID. That would be this one. That will be equal to, and here in this table where we have the gene IDs in the row names. So we can say row names. That's the BG table. So that will fix the problem. Yeah. Then if we want to remove this, and then we can put here BG table. Okay, let's run this. Then hopefully, yeah, that will fix the problem. As I said, yeah, still, I mean, well, as I was doing that before, I think that still will go fine because in some ways we were just using names. I can hear now my computer just do some complaints about this search. As I said, we can keep just as before. Okay. So I said, we can still keep this as it was, because in this case, I can see that we only wanted to have gene names. Okay, yeah. So let me go back here and still replace this by the original one. Okay, at least as I said, it's kind of giving you some idea of how we can change. If for some reason you need to stop this, we can try this. Okay, perfect. So let's run this again. And that will be much faster. Okay. So again, well, screen. So that is unique for that name. And then we want to retrieve the number of significant genes. So that is the command that we need to use. So what this command does is we are asking how of these result genes, how many of them have a p-value of less than 0.5. I guess that's the correct thing will be less or equal. So if we run this, it's telling us that we have 263 genes that met this requirement. Okay. If instead p-values, we want to use q-values, okay, remember that potentially we could use q-values, we have 0. Okay, so it means that in this case, when we are using the false discovery rate, we will not have significantly different genes. So still, we can suspect that these 263 genes that we are getting are false positives, because when we run a false positive correction, we get rid of those. At some point, also, false positive correction depends of the variance, so sometimes you want to still keep balance. Okay, so, and then we can plot the results. So to plot the results, I can copy and paste this. And this is an histogram, so we are plotting the result as a histogram. Okay. Of course, I think that there is some error here. Okay. Breaks, sig. Let me see. Yeah, maybe this was problem again. Okay. No missing. Yeah. No, it's depending on the, well, the thing is when you copy and paste commands, you always have these kind of problems, not because of when you pass from one font to another, you always have these kind of problems. So let me see, I should have the real command here. So let me copy and paste. Maybe that will be. Okay. Yeah, as I said, some place there we have a problem with a quote or something. So, okay, so that is showing us different number of differential expressed genes depending on the percentage. Keep in mind that this is the default discovery, the default change. So it means that these genes, you should not have genes here, but still, for some kind of reason, you have the statistical analysis told you that you may have some differential expressed genes here. In theory, you should have more here and here, but you don't see them. Then you have a peak very close to zero. That still will not be a good signal, okay? A good signal is that really you need to have, yeah, maybe in one condition more is twice expressed than the other, okay? So that will be 1 or -1. Okay, so that is, yeah.
I'm sorry. This is due to, I mean, this graph actually represents non-adjusted p-values, so multiplicity is not taken into account here, right?
No, yeah.
Okay, and you can still do that and present the results, or you usually have to use q values instead of p-values. I'm really a little bit confused about that. I mean, when you should use p-values and q values instead?
You always should use q values, except that using q values doesn't give you a differential expression.
Oh, okay.
So the thing is, so this is the point. Q values, so RNA-seq analysis from a statistical point of view is going to give you a lot of false positives because you don't have enough replicates. So this is the problem, no? We are playing with 3-5 replicates maximum, so you don't have enough replicates. So when you run a statistical analysis, no matter what, because you don't have enough replicates, you are going to have a lot of false positives by the nature of the data that you have. So in order to correct that, you can use this false discovery rate, that is this Q value that you see. And of course, you should always do that. well, the thing that happens is that sometimes the variance in your samples, let's say that if the R values that you have, when the R square values that you have are not good, it means that you are going to have a lot of variation between your samples. And when you run this analysis, you are not going to have almost any gene that is going to be under the Q value of 0.05. So in this case, I'm using p-values because of that. Other example, when you can have p-values is when you are working with trees and it's difficult to have good biological replicates. So still the differences you're going to see are much that at some point these Q values are not going to be useful because you have too much variance between the samples. But as I said, so my suggestion in this case is always use Q values. And if you run Q values and you have 0 differential expressed genes is when you need to think to use P values.
Okay, perfect. I get it. Thanks.
Yeah. Okay, so that is how it should look like. And then we want to generate a table with the results. So as I said, in this case, we want to add means and the standard deviation, mean and standard deviation for everything, and then write everything into a table. So to do that, yeah, you can, let me go here and can have these things here. So we can run all these commands. So if we run this, well, if we run this, this is going to give you as an error. Let's forget the G. Okay. So yeah, so that will be for the differential expression. Questions that you may have about this part. Okay, so if you don't have any other question, let me move to the DSEC. As I said, you have, this is the flow that we are using for this analysis. You have a good tutorial here that you can follow. And the first, of course, is to transfer the HTDSEC count here. So the thing I'm going to do now is, okay, let me save this. I'm going to open a new R script, and then I'm going to clean this one. Clear console, clean history, and clean plots, and move to files. So the first thing is I need to copy everything. So I have copied everything in this HTSEC count DSEC, and here I'm by gene. So I have all the samples here. So yeah, so that is the counts, as you can see here. So we are going to first set this as a working directory. We are going to just use this to move, to start this other script. Okay, so that is the previous step. The next is we are going to call the library, DSEC2. This is another example of, you can call the library or you can set up the working directory. No, first, that is not a problem. And then we want to create these sample files. Okay, so let me copy this here. So we want to generate a table, a Fino data table, like the one that I presented to you before. But we are going to do this in a different way. So we are going to do everything R. No, we are not going to open any text file, and we are going to play with that. So the first is so we are in the same directory, not the files, and we want to grab only the files that contain the I2TA word. So these are the files. So we want to put this in this sample file vector. Then we have all the conditions. So how we have been selecting the conditions is we know that C24 only is pure lines, as is Columbia. And then we want to have the rest. By the way, just I'm seeing, yeah. So it's important that these sample conditions fit in this. So I can see that now I have C24, 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 Columbia, 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 and then Columbia, 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 and then Columbia C24. So in this case, the sort is different. So probably I will need to change here this. So these hybrids are going to be here. Okay. So now we have the sample conditions as it should. So let's run this. Okay. Now we want to create the sample names. So we want to remove the I2TA and this HT count from the same names that the sample files. We do it. And then we want to create this table. Okay. Let's check the table. So the table sample names, so let's see that pure lines fit with the thing that should be pure lines, and hybrids fits with the thing that should be hybrids. Okay, perfect. So it looks like yeah, we have that. Okay. Next, yeah. And then we need to factor the condition, create a factor from the condition. So let me go back here. We cannot forget do a factor. Factor is a special. So what is a factor? Is a special vector in R that you have levels, no? So basically, you say that you have two levels, that is hybrid or pure line, and then it tells you which data do you have, no? But you have different levels. Okay. So that is for the preparation. Data upload. The data upload is simple. So we use this function. So let's go to exercise 2.1. So this is the function. I think that we have the quotes where we should. So let's upload the data. Okay. So let's check what do we have here. So DDSH sec. And this gives us information about what we have. So we have 33,341 genes. All the row names looks like gene names, and then the call names looks like the sample names. Okay. To get some stats, that is one, or then we can use summary of counts. So we do the summary of counts. This is how it looks like. So keep in mind that we are looking, in this case, to not FPKMs, but counts, and you look that the counts looks more or less the same, okay? Okay, so that is to data flow. Data filtering. For data filtering, we are using, because we are using counts and not FPKMs, we are using a different criteria. So we are going to keep only the genes that has a summer a sum of 10 or more, okay, of counts. So we run this command. Then I forgot something. Oh, okay, so we run this, run this. So now, for example, if we type ddshtsec, it tells us that we have 23, not 33 as before. And if we run summary, you will see that summary has changed and the mean now is higher, okay? Is 227, for example, in the first case. Okay, so that is for filtering. Then go to the differential expression. Remember that we said that in this case, we are going to calculate the differential expression first. So we run this dissect function and then results. So first, let's run the dissect function. And then we want the results. Okay, results. And now we want to check of these results, how many different differential expressed genes you will have. And let me see. And then, sorry, summary of test. So first, this table. This is telling us that we have 307 differential expressed genes, so we have a little bit more. And then the summary tells us that, well, when we are looking for adjusted p-value, we have indeed 3, I think. Yeah, we have 3 differential expressed genes. So let me see, not p-value, but p-adjust. Yeah, we have 2, okay, with p-adjust. So in this case, even potentially we have 2. But remember that it doesn't look good because we have this problem with the replicates. Okay, so that is for differential expression test, and then the quality control. So for the quality control, first is to generate this plot map. So let me go here. This plots exercise 2.4. So we run this. This is how it looks like. And you see this red. Okay, so these reds are telling me that I have, in this case, by default, the adjusted value that they use is not 0.05, but 0.1. So look into that, we have 3. So you can see these 1, 2, and 3 red dots here, and then you can see the mass of genes here. So that will be with the map plot. Sorry, this is not a window. Then let's compare for one gene that contains the lowest p-value. We can compare with the lowest p-value. Also, we can compare with the p-adjust value. So let's start with the p-value. So if we do this, this is called the gene. Okay. So in theory, with the p-value that we really have something different between the pure lines and the hybrids. Or even, as I said, if you want to get the p-adjust, probably will be the same. You have a low p-value. You also have a low p-adjust value. No, it's not the same. But still, you can see, no, you have with these normalized counts, you can see that this also has significant, no, will be significant. Of course, this can happen by random. Okay, we have 20,000 genes, so one of them will still met the conditions, even if the conditions are not good. Okay, so that is plot counts, and then heat map. So heat map, first we need to transform and do some normalization steps. So the BST is a normalization step. Then we calculate the distances. We are calling two libraries. We want the R color viewer. Well, we already have that, but also we can call feed map, and then we can transform this into a matrix, put some names, and then finally write the feed map. So. And of course, this is telling me that I have problems with double quotes. Okay, so it looks like we're done. On this. And this is how it looks like. And still, even with the thing that we fixed before, we see that replicate ones clustered together, like in the other case. And then we have two replicates 2, 2 replicates 3, that cluster together. So you can see here, no, the clustering. So yeah, again, this doesn't make much sense because the samples doesn't cluster together. Okay, so that is all for the differential expression analysis. Questions that you may have about this part?
Yep, I have one.
Okay.
A very basic one. So what is the output of the sec2? I mean, I can obtain the list, a written list of differential expression genes.
Well, so you want to get the list
Yep.
here? Yeah. So here when you go to this option, so you have res, no? So res is head. So res is the results, no? So this is all of the results, and here you have the genes. So you want to have the differential expressed ones. This is a data frame, so you can say res, and then as a data frame we are going to select only the rows. Let's go to the p-adjust value.
Okay. Yeah.
Of 0.1, as we will have 3, no? So if we do this, sorry, contains what is? Let me see. What is the problem here? Yeah, maybe res is not a data frame. No, it is. Let me see. Res. We adjust 4.1. Yeah. Okay. So now, being a data frame, okay.
You could try with a which function, maybe.
Which function? Yeah. All right. So. Yeah, usually don't use. Yeah. So yeah. So that will be which function. So now with the data frame. Yeah. So that's how you get the ID, the gene ID.
Oh, okay.
Of course, if you want to, you also can use merge to merge this with expression. You want to have also the expression values.
Okay. Okay, thanks.
Yeah. Okay. So, any other question?
I do have an additional one. Actually, could you go a little bit into detail as what the dist TSA VSD function does? Because it's a little bit tricky to me.
TSD.
It's the second line of the last command section. The sample dist equals dist TSA VSD. I mean, I can't really read that.
Can you give me the number?
Yeah, sure. It's 32, line 32.
32. Okay, this one. Okay, so this is the distance function. This is a T is transposing, and assay is a function to test this BSD. So let me see if I don't remember back. Assay. So yeah, summarize experiment class like a matrix. So in other words, so this BSD is the normalized. This is a transformation with a normalized. So the thing that you do is you transform this, your original object that you have, with the expression is you normalize by some specific function that they have. So when you apply assay on BSD, the thing that you do is you get basically a summary with all the expressions. So the thing that you do is you extract from this BSD object that contains a lot of different information, because it's not a data frame, but it's an object with different slots. You obtain, basically with assay, you obtain the expression. And once you get the expression, because the distance in this case need to be, I don't know why you need to have this transposed, but the thing is that in the protocol that I was looking, in this case, you transpose the matrix. Okay, so that will be the case. So you have columns, you transpose rows by columns, and then you calculate the distance. So basically, it's the distance between each of the samples.
Okay, thanks for breaking it down. Thanks a bunch.
Yeah, as I said, assay will abstract that, but I think I will imagine that with BSD, oh yeah, because you only have conditions and factors. Conditions and size factor. Yeah, you need to use assay to extract the expression. Okay, any other question? Okay, so you don't have any other question. I'm going to stop the recording first, and then we'll go for stop recording.